or isoprotein can actually have a lot of different variants or forms, leading us to the term isoform. So an isoform of a protein is just a variant or a version of a protein. And so it carries out the same overall function, but it has some different properties. So maybe it works faster or slower. It has different regulatory properties. You might have an isoform that is made in your liver and an isoform that's made in your brain and one that's made in your muscles. And they have slightly different properties that allow them to better suit the tissues they're in. You could also have isoforms in your mitochondria versus in your cytoplasm. You might have isoforms that are made at different points in life. So there are a variety of different reasons why you might want to have these different isoforms. They can come from the same gene or they could come from different genes. And so some of them come from different genes. So you have an ancestral gene that then gets duplicated and now you've got two copies of the gene, and then they can evolve separately. So you have the same kind of overall protein doing the same sort of thing, but it can do them in slightly different ways because you've altered the sequence a little. Maybe you've introduced a regulatory site. Now you can have phosphorylation, different properties. And so those could come from separate genes. We would call those paralogs, which are a type of homolog. You could also, however, get these isoforms forming from a single gene that just gets processed differently. So if we think about a gene, we've got a DNA sequence and we have exons, which are the regions that actually have the information for putting together the amino acids, interspersed with introns. And in the processing of the pre-messenger RNA transcript that gets made from that DNA, you cut out the introns and splice together the exons. You can do this in different ways, and then you can therefore be kind of like skipping exons, including different exons, and that sort of thing. And so this gives us alternative splice products, and those alternative splice products could then, if they're functional, produce different protein isoforms. You could also get things like alternative promoter usage, and so you can have the RNA polymerase, when it's making that pre-mRNA copy, it could actually be starting sooner, and then you get a longer product, or later and you get a shorter product in your messenger RNA. And then the messenger RNA itself, the ribosome can actually start at a different site, a different start codon, so you could have alternative start codon usage, which would then influence the final protein that gets made as well. So those will all be getting made from the same gene. A great example is pyruvate kinase, which carries out the last step of glycolysis. We have 4 versions of it coming from 2 genes. The PKLR gene gives us the liver and the red blood cells, and those are made from different promoters. And then the PKM gene gives us PKM1 and PKM2, which are made from alternative splicing. And those have different properties that allow them to be regulated differently.